in East Africa. So I'd like to start by thanking my funders and collaborators and move straight on to talk about two East African soda lakes found on the border of Tanzania and Kenya. The soda lakes Natron and Magadi, which are sited in a shallow basin in a highly volcanic region. And they represent an extreme aquatic environment. Each of the lakes is covered in a trona crust of sodium carbonate precipitates that covers the majority of the lake surface, but bordered by open water lagoons that are fed into by volcanic springs. The springs themselves are depauperate in fauna and flora, very few invertebrate species, no vascular plant species, but a wealth of endemic cyanobacterial species. They're incredibly extreme for aquatic organisms, having high temperature, high salinity, high pH, and low oxygen levels. But there is a single genus of cichlid fishes inhabiting these springs and the lagoons, and that is the Alpolapia cichlids, that are a subgenus of the Oreochromis tilapine cichlids. And they exhibit a range of physiological adaptations to this extreme environment. There are four species within the lakes, two species here with terminal mouth positions that's characteristic of the rest of the Oreochromis clade, and two species with subterminal mouths. They're found in fragmentary populations around the border of the lake, and there's variable levels of gene flow between those populations. And we're, for this study, also considering the putative sister group, Oreochromis amphimalis, which is found in less extreme soda conditions throughout Tanzania. So genomic analysis of rad data demonstrates shallow molecular diversification across the whole Alpolapia radiation, less than between populations of the outgroup, and there's evidence of ongoing gene flow between the species in Lake Natron. Using that genomic data and comparing it to morphological data of geometric morphometric analysis, we see that the molecular distance shows a positive correlation with the morphological distance, and interspecific comparisons up here are more distance than the interspecific population comparisons here. But when we add in the outgroup to that comparison, we see that the molecular distance of the Alpolapia scales right back, and we have extensive morphological divergence, but very little molecular differentiation, which may be explained by the observation of heterogeneous genomic differentiation with low levels of FST across the genome, but narrow peaks of high FST, which is in contrast to the comparison with the outgroup showing homogeneous high levels of differentiation across the whole genome. And we do see a phenotype-environment correlation based on morphology with ecological data of stable isotope data, but that is restricted entirely to the carbon isotope, and differentiation between the species is based on resource type and foraging mode, but not on trophic level, and all the species have diversified as herbivores. We don't see any specialized invertebrate feeders or fish, fry, or egg scale feeders that we see in other small-scale fish radiations. And one reason for this, if it's possible to play the video here, may well be specific to the soda lakes, and that is the really extreme environment requires these fish to maintain a high metabolic rate. Fortunately, it doesn't look like the video is going to play, but they have very high metabolic rate to cope with the osmotic pressure imposed, and it may be that the productive environment of the soda lakes constrains all that diversification to only occur along the herbivorous axis, and it's too costly possibly to cross the fitness landscape to another trophic level. So just to summarize, it's an incredibly recent diversification of cichlid species with high morphological divergence, but very shallow molecular divergence within these species. There is heterogeneous genomic differentiation across the genome, but the ecology is differentiated solely along a herbivorous axis, which could possibly be explained by metabolic constraints within an extreme environment. Thank you.